السلام عليكم ورحمة الله تعالى وبركاته. I'm Salma Al-Mahdi, and today is the first lecture in fatty acid metabolism. We will be discussing fatty acid synthesis. We will define what is meant by fatty acid synthesis, and we are going to go through the physiological roles of fatty acid synthesis in the body, identify tissues and cellular compartments where fatty acid synthesis occur, and we are going to describe the enzymes that are involved in the process, including acetyl-CoA carboxylase and fatty acid synthase. We are going to list the steps of palmitic acid synthesis and identify the substrates and cofactors that are required, and we will describe the integration that is needed to provide acetyl-CoA and NADPH for lipogenesis. Finally, we will describe how fatty acid synthesis is regulated. Fatty acid biosynthesis, also known as lipogenesis, is an anabolic process concerned with conversion of surplus glucose and intermediates such as pyruvate, lactate, and acetyl-CoA to fat. So it is a building up process, an anabolic process. The body converts the energy and metabolites and carbohydrate molecules that are more than needed, the surplus, to fat stores. And as you know, a fatty acid biosynthesis role is the same as for lipid biomolecules that you have discussed in the lipid chemistry course, as energy storage in the form of triacylglycerols and triglycerides. Lipids are needed for membrane biosynthesis, particularly phospholipids, and for the synthesis of bioactive lipids such as prostaglandins and leukotrienes, for thermal insulation, as you know, and for mechanical protection of internal organs, as precursors for complex molecules such as lipoproteins and complex lipids, for the structure of the brain and the function and neural signaling, and storage of lipophilic compounds such as lipid soluble vitamins, and for the adaptive response to fasting and feeding. Where does fatty acid synthesis takes place? At the level of the tissue, fatty acid synthesis occurs in the liver, in the kidney, brain, lung, mammary gland, and adipose tissue. Intracellularly, fatty acid synthesis occurs in the extramitochondrial compartment, including the cytosol, where the complete synthesis of palmitate occurs, and in the endoplasmic reticulum, where the fatty acid chain elongation occurs, and particularly the smooth endoplasmic reticulum. Fatty acid synthesis is carried out by three enzymes. Actually, they are called enzyme systems because each one of them has two or more catalytic activities, so it is a multi-enzyme. And these include acetyl-CoA carboxylase, fatty acid synthase, and fatty acid elongase. Acetyl-CoA carboxylase, this multi-enzyme protein, contains two catalytic activities, biotin carboxylase and a trans carboxylase, in addition to biotin. And as you know, biotin is a coenzyme for carboxylases. It also includes a biotin carboxyl carrier protein and a regulatory allosteric site. This is the structure of acetyl-CoA carboxylase. The red part here is a biotin carboxylase. The blue part is a trans carboxylase. The violet part is a biotin carrier protein. This small yellowish structure here is the biotin. Fatty acid synthase is a polypeptide that contains 7 enzyme activities. It is a majestic enzyme. It is a very big enzyme with several catalytic activities, up to 7 for each polypeptide. This polypeptide occurs as conjugated or linked units in mammals, yeast, and birds, but the units are separate in bacteria, plants, and others. It cannot, in mammals, the subunits cannot be subdivided without the loss of activity. So the structure here dictates function, and function is dependent on the structure. When the structure is disrupted for any reason, the function will be lost. This aggregation is beneficial. Why? Because it provides greater efficiency and it will freeze the reaction for interference by competing reactions, There will be no permeability barriers. All the products are sequentially present together, and at the level of the gene for these enzymes will have coordinated gene expression because they are next to each other. They are easily transcribed and translated with fewer inborn errors and genetic mutations. Fatty acid synthase is a homodimer. It contains two identical monomeric units. The two monomers slide head to tail. So this blue one here contains the 7 catalytic activities, lies in the opposite direction, head to tail, with the second monomer shown here in white. It contains 4-phosphopyrothene, the active form of vitamin B5, as well as cyanogen. The subunit division structurally is when the transversely. The one that is perpendicular to it is a functional division. So for the fatty acid synthase to function, these subunits here from this monomer will collaborate with those subunits here in the other monomers, and those is those. So the functionality crosses the barrier. Each part collaborates with the other part of the second monomer. Fatty acid elongase is present in the endoplasmic reticulum. The ELOVL, which stands for elongation of very long chain fatty acid, is a family of enzymes. They determine the length of both saturated and unsaturated very long chain fatty acids. A fatty acid elongase has 4 catalytic activities, as we are going to see. The layout of fatty acid synthesis is that our immediate substrate, the initial substrate, is acetyl-CoA that comes from carbohydrates, particularly glucose. The subsequent substrates are malonyl-CoA. The end product, as we are going to see today, is malmethane, but the body can synthesize whatever is needed in terms of length or the state of saturation, desaturation. But we are going to study the synthesis of palmitate as an example today. The requirements for fatty acid synthesis include NADPH as a reductant, ATP as a source of energy, manganese, biotin, or as a coenzyme for carboxylases, and bicarbonate as a source of carbon units or carbon dioxide. This is the structure of palmitate as you know it. 16 carbon atoms. This is carbon atom number 1, this is carbon atom number 2, the alpha carbon, this is carbon atom number 3, the beta carbon, and this is the last 16th carbon here, is the omega carbon. The synthesis starts, of course, by 2 carbon atoms that come from acetyl-CoA. We said it is the initial substrate. They will make the last carbon atoms of the full chain of the palmitate or palmitic acid. So the initial or the starting carbon atoms that came from acetyl-CoA will make up carbon atom number 15 and number 16. The rest of the carbon atoms will come from malonyl-CoA as a substrate, and any further additions or elongation of the lipid up to n number of carbon atoms also come from malonyl-CoA as a substrate, and they will be added by fatty acid elongase. We need NADPH for reductive synthesis of fatty acids. The sources of NADPH are 3. The main source is, of course, the pentose phosphate pathway, and that is why tissues that are active in lipogenesis are also active in pentose phosphate pathway. Moreover, both pathways, as you know, are cytosolic, so there will be no permeability issues regarding the transfer of NADPH. A second source is malic enzyme, also called NADP malate dehydrogenase. A third, however, non-substantial source of NADPH with extra mitochondrial isocytrate dehydrogenase. The first reaction in fatty acid synthesis, formation of malonyl CoA. This is a two-step reaction. First, the biotin or part of the fatty acid synthesis is carboxylated, and the source of the carboxyl group is bicarbonate. ATP is needed for energy. Then the carboxyl group will be moved by transcarboxylase to the enzyme to the acetyl-CoA to form malonyl-CoA, which is a 3-carbon atom compound. The second reaction is formation of acetyl-malonyl enzyme, an enzyme, a fatty acid synthase, that is linked to both acetyl and malonyl residues. This one also occurs in a two-step reaction. And we are going to see how the two parts of the monomers collaborate. Here, this end is the thiol group of the cysteine in this monomer reacts with acetyl-CoA, while the other end reacts with the thiol group of the 4-phosphopantothene reacts with malonyl-CoA. This reaction is catalyzed by acetyl transacylase and malonyl transacylase. So this bluish structure here, 1 and 2, they refer to the two monomers of the enzyme. Acetyl residue is now attached to the ferrous monomer, while malonyl is now attached to the second monomer, and CoA of the two molecules is released. Now our product is acetyl malonyl enzyme. Acetyl and malonyl residues are attached to the enzyme. The third reaction is formation of an acetoacetyl or a 3-ketoacyl enzyme. The acetyl group here attacks the malonyl in the second monomer, catalyzed by 3-ketoacyl synthase. Carbon dioxide is released, and now we have a 3-ketoacyl or acetoacetyl enzyme. The newly added carbons are attached to the thiol group of the 4-phosphopantothene. The fourth reaction is reduction by 3-ketoacyl reductase, and ADDH is used as a reductant. And as you can see here, we have our product, the 3,3-hydroxyacyl enzyme. The fifth reaction is the dehydration reaction. By dehydratase, a molecule of water is removed, creating a double bond, and the product here is a 2,3-unsaturated acyl enzyme. The sixth reaction is another reduction, wherein ADDH is also used as a reductant. It is enol reductase, and the acyl enzyme is now formed. The double bond is now saturated. We have an acyl residue that is 4 carbon atom in length attached to our enzyme, so it is an acyl enzyme. The seventh reaction is a displacement, whereby a new malonyl-CoA molecule comes into action. It will displace the acetyl residue from this end here, from the attachment to the thiol group of the 4-phosphopantothene to be attached to the thiol group of the cysteine, and it will occupy this place, and now we have an acyl malonyl enzyme. From now on, there is repetition of the third to the seventh step 6 times to create our 16 carbon atom acyl or bimethyl radical. And the eighth reaction, of course, we need to separate our product, palmitate here, the acyl residue palmitate, 16 carbon atom, from the enzyme, and this is done by a thioesterase, which releases a free enzyme in addition to palmitate. The overall reaction for palmitate synthesis from priming acetyl-CoA molecule and several malonyl-CoA molecules, one for the initial action and the rest for the sixth repetition of the steps 3 to 7. We have acetyl-CoA here, one molecule of acetyl-CoA, 7 molecules of malonyl-CoA, 14 in ADPH, and the result is palmitate plus 7 carbon dioxide molecules plus 6 molecules of water plus 8 CoA plus 14 NADP. If you want to revise the number of carbon atoms, acetyl-CoA contains 2, malonyl-CoA contains 3, so it will be 3 by 7, 21 plus 2, 23, minus those released as carbon dioxide, minus 14 will give you the 16 carbon atom palmitate. What is the fate of palmitate from now on? Palmitate has 3 fates. Two fates. The major one being esterification, triacylglycerol and cholesterol esters. A second fate is chain elongation. If the body needs fatty acids that is longer than 16 or unsaturated, there will be addition of more carbon atoms by elongases and introduction of double bonds by the saturases. And before going into either pathway or either fate, palmitate has to be activated first by being converted to palmitoyl-CoA. CoA here signifies or indicates that this is an active molecule, like glucose 6-phosphate is the active form of glucose, palmitoyl-CoA is the active form of palmitate, and this is done by an acyl-CoA synthetase, whereby ATP is needed for energy and CoA is introduced into the molecule. So far, I would like you to know that there are 2 centers of activity in each dimer of the fatty acid synthase complex. They function independently, simultaneously. Those 2 molecules of palmitate will be formed from one fatty acid instance. And the primary acetyl-CoA molecule, as we said, forms carbon atoms number 15 and 16 of the palmitate. Addition of subsequent C2 units is via malonyl-CoA. Other priming molecules can be used as well. For example, we can start with butyryl, which is 4 carbon atom priming molecule, and propionyl 3 carbon atom compound. If the priming was with propionyl, the end result or the product will be, of course, a fatty acid with an odd number of carbon atoms. Let's do some integration here. We said the provision of acetyl-CoA and NADPH for lipogenesis requires integrated action of several pathways and enzymes, including glycolysis, pentose pathway, citric acid cycle, lipogenesis, tricarboxylate transporter, ATP citrate lyase, and extra mitochondrial isocytrate dehydrogenase. Let's go into the figure. These notes are for you to revise it later. This part of the figure here, the grayish part, represents the intramitochondrial compartment. The whitish area is, of course, the cytosol. We said acetyl-CoA for fatty acid synthesis comes from carbohydrates, particularly glucose via glycolysis. So this is all glucose molecule here, undergoes glycolysis, aerobic glycolysis in particular, to form pyruvate. Pyruvate can cross the mitochondrial membrane via pyruvate transporter. In the mitochondria, pyruvate will be decarboxylated to acetyl-CoA. Acetyl-CoA unfortunately cannot cross the mitochondrial membrane, and the solution is to cross it in the form of citrate. So acetyl-CoA will combine isoxazotate to form citrate. This is the first reaction, of course, of the citric acid cycle. Citrate now can exit the mitochondria to the cytosol And in the cytosol, citrate under the influence of ATP citrate lyase will reform acetyl-CoA and oxaloacetate. Acetyl-CoA now is available for fatty acid synthesis, for example, to form palmitate, while oxaloacetate can form malate with malate enzyme, and then with the malate dehydrogenase, it can go inside the mitochondria again to be part of the citric acid cycle. Let us revise the sources of NADP here. Pentose phosphate pathway, we said it is the main source. Malic enzyme here, which converts malate to pyruvate. It is oxidative decarboxylation as well, and it is a source of NADPH for reductive synthesis. The third pathway is the isocitrate dehydrogenase cytosolic pathway. So if you want to elongate the side residue, we need to employ the action of fatty acid elongates. Fatty acid elongation means conversion of an acyl-CoA to an acyl-CoA derivative that has two carbon atoms or have two additional carbon atoms in length. The fatty acid elongase, as we said, has four enzymatic activities. The activity of the elongase is abolished by fasting and diabetes. The substrate for elongation are saturated fatty acids that are 10 carbon atom in length or longer, and unsaturated fatty acid. In the brain, elongation of steroid-CoA increases during myelination to provide carbon to provide fatty acids that is 22 or 24 carbon atom in length for the synthesis of sphingolipids. The first step of elongation is formation of 3-keto-acyl-CoA. This is our acyl-CoA residue or fatty acid that we want elongated to add to its length. We are going to use malonyl-CoA as a source of carbon atom, and the influence of the 3-keto-acyl synthase activity of the elongase. CoA will be released plus 1 carbon atom, and the 3-keto-acyl residue here is formed. The black dots here represent or indicate the carbon atoms that came from malonyl-CoA, while the circles here with the empty core center represent the carbon atoms that come from the acyl-CoA. The second reaction of elongation is reduction using NADPH, and the enzyme is the 3-keto-acyl reductase activity of the elongase enzyme. 3-keto-acyl-CoA is reduced to 3-hydroxyacyl-CoA. Then this 3-hydroxyacyl-CoA will be dehydrated with removal of one molecule of water to form 2-trans-enoyl-CoA with introduction of a double bond, which will be reduced. NADPH will be used and saturate the double bond using a 2-trans-enoyl-CoA reductase. And now we have an acyl-CoA that is 2 carbon atom longer than the initial substrate. How fatty acid synthesis is regulated? The nutrition state is the main regulator of the rate of lipogenesis, and there is an inverse relationship between hepatic lipogenesis and the concentration of free fatty acids in serum. The rate of free fatty acid synthesis will be higher in the well-fed state, where you expect there is high concentration of dietary carbohydrates, and when sucrose is fed in a state of glucose. And here is a question for you. Why does fatty acid synthesis increases rate-wise when sucrose is fed instead of glucose? Let me know your answer. On the other hand, fatty acid synthesis is depressed when there is high concentration of free fatty acids in plasma, which occurs in conditions where there is restricted caloric intake, high fat diet, and an insulin deficiency, for example, in diabetes. Mechanisms of regulation include short-term mechanisms such as allostery and covalent modification, and long-term mechanisms by induction or repression of enzyme synthesis. Allostery or allosteric effectors here include citrate, which is an allosteric activator of acetyl-CoA carboxylase. Citrate, of course, signifies energy abundance. It increases in the well-fed state, so it will activate acetyl-CoA carboxylase. It is a well-fed state, there will be surplus carbohydrate, all of which can be shifted to acetyl-CoA to be acted upon by acetyl-CoA carboxylase and fatty acid synthase, and to store it as fatty acids or lipids. Acyl-CoA, on the other hand, is an allosteric inhibitor of acetyl-CoA carboxylase. Acyl-CoA, of course, is the product, so this is a form of negative feedback for the enzyme. Pyruvate dehydrogenase regulates the availability of acetyl-CoA for lipogenesis because it synthesizes acetyl-CoA from pyruvate. There is an inverse relationship between free fatty acids and the proportion of active pyruvate dehydrogenase. Regarding the covalent modification, insulin via the action of a phosphatase leads to activation of acetyl-CoA carboxylase, and on the same time, insulin inhibits lipolysis, so it will decrease the concentration of free fatty acids in serum, so this will de-inhibit acetyl-CoA carboxylase. Glucagon and epinephrine, on the other hand, antagonize insulin by inhibiting acetyl-CoA carboxylase through a kinase that will lead to phosphorylation of acetyl-CoA carboxylase. As you can see here, the active form of acetyl-CoA carboxylase is a dephosphorylated form, while the inactive form of acetyl-CoA carboxylase is phosphorylated. So insulin, which is produced in response to high glucose following dietary ingestion or parenteral administration of glucose, this insulin will activate the protein phosphatase. This phosphatase will remove the phosphate group and activate acetyl-CoA carboxylase. So under influence of insulin, there is covalent dephosphorylation of acetyl-CoA carboxylase, and it is activation leading to active fatty acid synthesis. On the other hand, glucagon via cyclic AMP will activate a kinase that will phosphorylate and inactivate acetyl-CoA carboxylase, leading to inhibition of fatty acid synthesis. Induction and repression, of course, at the level of the gene for the two enzymes, fatty acid synthase and acetyl-CoA carboxylase systems. They are both adaptive enzymes because they adapt to the body's physiologic needs by increasing in the fed state and decreasing in fasting. Insulin induces their synthesis, while glucagon represses their synthesis. Here is an assignment for you. Look for the molecule that is called CHREBB and SREBB in the context of fatty acid biosynthesis, just to give you a glimpse of how the process has so many details. What we have discussed here is just the outlines of fatty acid regulation. Hint for you, BB is binding protein. For both molecules, I need the definition, the target genes, the function, activation, and the role in synthesis. Disorders of fatty acid synthesis can lead to a variety of metabolic dysfunctions, including energy deficits, abnormal lipid profiles, organ-specific damage, and uh, obesities of nature, variable nature of extent of obesity, and of course, in insulin diabetes type 1, there is inhibition of lipogenesis because of the deficiency of insulin. Deficiency of acetyl-CoA carboxylase can manifest as developmental delay, intellectual disability, and seizures. Fatty acid synthase deficiency can manifest as hypotonia, hypoglycemia, and cardiomyopathy. Manifestations of C-hydroxycyl-CoA dehydrogenase and enoyl-CoA reductase are the same, including developmental delay, intellectual disability, and seizures. This will be all. Thank you very much.